DNA fingerprinting. DNA fingerprinting is also known as DNA profiling. It is a technique used to identify individuals based on their unique DNA pattern. It was developed by Sir Alec Jeffrey in 1984 and it is widely used in forensic science, paternity test and genetic study. First, the sample is collected. The sample can be blood, hair, skin, cell or saliva. So, these samples are collected. First, blood sample is collected, then DNA is isolated from the cell using a chemical or mechanical method. Then these specialized enzyme called restriction enzyme that cut the DNA into fragment at a specific sequence. Then the DNA fragment are separated based on their size using an electric field in the gel, and then the separated DNA fragments are transferred to a nylon or nitrocellulose membrane, and then radioactive or fluorescent probe binds to the specific DNA sequence and the unique pattern of band is visualized under UV light or X-ray film. Create a DNA fingerprinting. There are applications of DNA fingerprinting are identifying suspect or victim in criminal investigation, paternity or maternity test, establishing biological relationship, and medical research, identifying genetic disorder or studying genetic diversity. And in wildlife conservation also, tracking genetic diversity and poaching investigations. These are some applications of DNA fingerprinting. Principles. The unique DNA pattern. The DNA is of every individual contains some unique sequence called repetitive DNA, that is the short tandem repeats or STRs. Then polymorphism. Difference in the length of these repetitive DNA sequence create a unique pattern forming the basis of DNA fingerprinting. Restriction enzyme and electrophoresis. These are the tools that separate the DNA fragment, allowing the unique pattern to be visualized. Then step by step process we have seen. First, the sample is collected. The blood sample is collected and it is carefully stored to prevent contamination. Then DNA is extracted from the sample using chemical lysis buffer or mechanical mechanism. Then the pure DNA is isolated. Then these enzymes cut the DNA at specific recognition site and then DNA fragment into small pieces of varying length. Then gel electrophoresis is done. The DNA fragments are placed on the gel using agarose and subjected to an electric field. Then small fragment move faster and farther than larger one. The small fragment move faster as compared to the larger one. So the DNA fragments are separated based on their size. Then southern blotting. What happens here? The DNA fragments are transferred from the gel to a nylon or nitrocellulose membrane. These steps ensure the fragments are fixed and ready for analysis. Then radioactive or fluorescent labeled DNA probe are added to the membrane and these probes bind to a specific DNA sequence. The probe highlights the repetitive sequence which form the unique DNA pattern and the membrane is exposed to the X-ray film or UV light. A banding pattern or the DNA fingerprint is visible. Then we have seen the application of fingerprinting. Forensic science. It is used to identify suspect by matching DNA for crime scenes with individual. It helps the identifying remains in disaster. In paternity and maternity test, established biological relationship between parents and children. And in medical applications also, it detects the genetic predispositions to diseases. It helps in organ transplantations to ensure compatibility. And in wildlife and conservation, it identifies poached animal and illegal wildlife trades. Monitor genetic diversity in endangered species. Then in archaeology also, the studies ancient DNA to understand the historical pollutions. Advantages. High accurate and reliable and can be performed within a minute quantities of DNA. Non-invasive sample collection is possible. Limitations. Expensive and time consuming. Required expertise and specialized equipments. Contaminations can affect the results. Thanks for watching. Do like, share and subscribe.